Edvotek instructional videos. Separating proteins using SDS Polyacrylamide Gel Electrophoresis, also known as SDS-PAGE. For this experiment, you will need protein samples, tris-glycine SDS electrophoresis buffer, a vertical polyacrylamide gel electrophoresis apparatus, a micropipette and gel loading tips, a power supply, and a precast polyacrylamide gel. Step 1. Boil the samples for 10 minutes to denature the proteins. Step 2. While the protein samples are boiling, place the precast polyacrylamide gel cassette into the electrophoresis unit. Follow the directions provided by the manufacturer to ensure that the gel cassette is properly oriented. Step 3. Add the 1x tris-glycine SDS electrophoresis buffer to the chamber. The sample wells and the gel cassette should be completely submerged. Rinse out the wells using a small transfer pipette or micropipette. Step 4. Carefully remove the protein samples from the boiling water bath. Collect the sample at the bottom of the tube by centrifuging or by gently tapping the tubes on the tabletop. Step 5. Load 20 microliters of the protein samples into the wells in consecutive order. Step 6. Place the safety cover on the electrophoresis unit. Connect the leads to the power supply. Set the power supply to the required voltage and perform electrophoresis. Refer to the experimental instructions for time and voltage guidelines. Step 7. Electrophoresis is complete when the dye front has run to the bottom of the gel. Turn off the power supply and remove the gel cassette from the electrophoresis unit. Place the gel cassette flat on the lab bench with the large plate facing down. Step 8. Using a spatula, carefully separate the two plates of the gel cassette. Place the spatula in the slot at the top edge near the sample wells. Twist the spatula to separate the two plates of the cassette. Repeat until the plates are completely separated. Step 9. Gently lift the front plate away from the larger back plate. In most cases, the gel will stay on the back plate. If the gel partially sticks to the front plate, let it fall onto the back plate. Step 10. Proceed to staining the gel or to Western blot.